Hey everyone! Today we will be talking about chromosomal mutations. So, what are chromosomal mutations or aberrations? These are modifications in chromosome number or structure itself. Various types of abnormal chromosome numbers include variations in the number of chromosomes, and these can also be classified as structural aberrations of the chromosome itself. So, what is the difference between aneuploidy and euploidy, right? So these two terms are terms that are under alterations in chromosome number. So aneuploidy involves the diploid number plus or minus an X number of chromosomes. So in this case, you have 3 chromosomes at chromosome 21, right? So in this case, an organism gains or loses one or more of the chromosomes, but not a complete set. Whereas in euploidy, you have multiple of the complete sets of haploid chromosome numbers. It's typically lethal in animals, but you can see it a lot more in plants. So in this case, this would be an example of euploidy because you have more than one set of chromosomes, okay? So another set of terminology for variation in chromosome numbers involve various types, categories under aneuploidy and euploidy. So as we said, aneuploidy is the explanation for that is the diploid number plus or minus X number of chromosomes, right? So in this case, one of the first ones would be monosomy, which is 1 minus the diploid number minus 1. So if you're like humans that have 46 chromosomes, a monosomic individual would have 45 chromosomes, right? So disomy is just a regular individual diploid. You would just have the regular diploid number. Trisomy, you have one extra chromosome, so than the diploid number. Then it goes tetrasomy, pentasomy, which just adds on one chromosome to each higher level that they go to. So tetrasomy is 2n plus 2, pentasomy is 2n plus 3, and then so on and so forth. It just continues up the number of chromosomes, okay? Then we have euploidy. Euploidy involves the multiples of the haploid number, right? N. So diploidy, as we know, it's 2n. Polyploidy is anything over 2n, so that would be 3n, 4n, 5n. 3n is also known as triploidy, and then tetraploidy and pentaploidy are like 4n, 5n, so on and so forth, okay? Then you have autopolyploidy, multiples of the same genome. Allopolyploidy, it involves multiples of closely related genomes. So it's just a matter of distinguishing between which types of genomes are involved, okay? So that's the terminology involved, right? Now we're gonna go into a question that involves using this terminology. For a species with diploid number of 40, indicate how many chromosomes will be present in somatic nuclei of the individual that are haploid, tetraploid, trisomic, and monosomic. So with haploid, we know that, so what do we know about haploid? So we have the diploid number. Diploid number is double the haploid number, right? So we would do the 2n value. This is supposed to be 2n divided by 2, which is 40 divided by 2, right? So then you have 20, right? Whereas with tetraploid, it's 4 times n, right? 4 times the haploid number. So you put 4 times 20, which is equal to 80, okay? Then you have trisomic. Trisomic just means that you have one extra chromosome. It doesn't matter which one it is, it's just trisomic. So in this case, it will be 2n, so 1 plus the diploid number. So when you look at that, you see 40 plus 1, which you end up with 41 chromosomes. With monosomic, we know that it's 1 less than the diploid number. So it's 2n minus 1, which is 40 minus 1 equals 39. So this is kind of the process you would go through to solve these questions. So you have this terminology. Just make sure you know which one goes with what, okay? So when you think about all of this stuff, how do these variations actually occur with the chromosome number? So how do these variations occur? So these variations occur through a process called non-disjunction. It occurs in either meiosis 1 or meiosis 2, right? So in this figure here, when it's the first division and it ends up with non-disjunction, you have these guys were like their disjunction, and then they end up with no chromosomes here, but then comes down, it ends up monosomic when it comes to pairing with another genome, right? When the homologous chromosomes come together, and then it ends up here. When it comes into the second level of meiosis, then you see this goes normal disjunction and comes down pairing up properly. In the second non-disjunction, you see the chromosomes getting trisomic and monosomic, right? So that's how these variations end up occurring. So what is a karyotype and why is it useful? So a karyotype is essentially a complete chromosomal set of the nucleus of the cell during mitosis. And it is represented with all pairs of chromosomes arranged in the order of size, which is the ideogram. This is a karyotype. You can see the different chromosomes. Why is it useful? It allows you to diagnose different chromosomal abnormalities within utero, and then you can analyze genetic diseases because of the whole aneuploidy situations and missing pieces and translocations. So now that we understand what that is, we're going to talk more about the different examples of chromosome alterations. So some examples include deletions and duplications. These are the types that cause the total amount of genetic information to change within a chromosome. These actually are more likely to result in a phenotype. The next set we have is inversions and translocations. The genetic material, the amount itself is the same but rearranged from the regular setting. So the affected individuals in this case will likely to be phenotypically normal and might have abnormal gametes, okay? So let's go through these different alterations. So what is a deletion? Deletion, as the name suggests, it's missing regions in a chromosome. So if you have a chromosome here, you have A, B, C, D, E, F, G. Here, when you have a situation where a deletion occurs, it could be like A, B, C, E, F, G, right? So D kind of gets lost when these occur. So in this case, deletions occur when chromosomes break in one or more places. The location of deletion can vary depending on what is going on. So a terminal deletion would be like near one end of the chromosome, or it can be within the chromosome itself. So that would be called intercalary deletion. So the interior of a chromosome itself, okay? So that would just, a deletion loop comes in, it breaks off and just shortens it out, and this part is lost. Okay, so now we have a question. In a homologous pair of chromosomes, one member is normal and the other has a deletion. How would these homologs pair during synapses? So when you look at this question, you have two homologs, right? So these homologs are, so we have one normal chromosome and one homolog that has the CD area deleted, right? So what happens is that when this is happening, whichever sections can actually pair up, and then the remaining section that has no homolog will end up looping out like this to form a deletion or a compensation loop. So this guy will loop out and try to pair up with its homologous bands itself. Okay. So what is a duplication and how did they arise? So duplication, as it suggests, it's a repeated segment or duplicated segment of a chromosome. It can be within the genome itself. So you have B and C here, right? So just one. But then when you have duplication, you have B, C going on like that. Okay? So the single locus is present more than once in the genome. So that's essentially duplication. And it typically arises when unequal crossing over between the synapse chromosomes occur during meiosis. So essentially, when unequal crossing over occurs, you end up with multiple of the same sequences or segments on the same chromosome. Okay, so describe what an inversion is. So what is an inversion? Inversion is essentially a linear gene sequence rearrangement. So essentially it is like flipping a DNA sequence so it detaches, flips, and then reattaches itself, right? So if you have BCD here, an inversion of this guy would end up DCB, okay? This is, as I said, it's a segment of the chromosome turned 180 degrees within a chromosome. There is no loss of genetic information because all of the genes are still within that chromosome. It's just flipped around. It typically requires 2 breaks in the chromosome, and reinsertion ends up inverted because it loops around. It can arise using chromosomal looping. So there are two types of inversions. One is called a paracentric inversion and a pericentric inversion. So in paracentric, it does not change the lengths of two arms of the chromosome. So, and the centromere is not part of the inverted segment. So so para as in around, it's nearby but not completely around it. Okay, so it's like here away from the centromere. So in this case, you have BCD. Here you have DCB, right? And it's away from the centromere. So that is paracentric. Pericentric, so that's away. So yeah, so BCD, DCB, everything switched around. So pericentric does change the lengths of the two arms of the chromosome. And the centromere is clearly involved with the segment. So for this example, you can see here, you have BCD around the centromere, and when it inverts, it also changes the arms of the chromosome, okay? So next thing we have is translocations. Translocations alter the location of chromosomal segments in the genome. So there are two types of translocations. One is called non-reciprocal, and the other one is reciprocal translocation. So in non-reciprocal translocation, you have one-way transfer of a chromosomal segment to another chromosome. So you have these two chromosomes. When non-reciprocal translocation occurs, you have a transfer from here to there, and it's staying that way, right? Because it's a one-way transfer, okay? Reciprocal goes both ways. So it involves the exchange of segments between two non-homologous chromosomes. And genetic information is lost or gained. It also has unusual synapses configuration during meiosis. So again, non-reciprocal, one way, reciprocal, two ways, okay? So this is a question. A woman is found to be heterozygous for a chromosomal arrangement between the second and third chromosomes. Her chromosomes are diagrammed here. So this is the diagram that they have for this woman who is heterozygous for a chromosomal rearrangement. What kind of chromosomal aberration is shown here? So when we look at these questions, what do we see? We see a two-way transfer, right? So that means it is reciprocal translocation. And so the next question is, explain the difference between the two translocation methods. And you have, as reciprocal translocation, you have the exchange of genetic material between non-homologous chromosomes, whereas non-reciprocal translocation involves one-way transfer of a chromosomal segment to another chromosome. Okay? So this is another question. So in a recent cytogenetic study on 1,021 cases of Down syndrome, 46 were as result of translocations. The most frequent was symbolized as T parentheses 14 colon 21 close parentheses. What does this actually represent? So when we look at these symbols like this, one of the things you should notice that the T in this case stands for translocations. So it's a translocation. So essentially this is what was happening with this case. And it's saying that this was the case because you have a translocation going on is with the chromosomes itself. It was just showing an even more um, genetic material on chromosome 14. So we can say that this symbol represents translocation between chromosomes 14 and 21. How many chromosomes would you expect to be present in T14 semicolon 21 Down syndrome individuals? We would see 46 chromosomes, right? Because you are not changing the amount of genetic material that is present. Okay, so to summarize the structural changes in chromosomes, you have deletions, you have missing regions, duplications, you have more single loci, locus is present more than once in a genome, rearrangements, inversion, it involves um, inversions involve a 180 turn in the chromosome, non-reciprocal one-way transfer of genes. Reciprocal translocation is a two-way between homologous and non-homologous chromosomes. Okay, so that's the summary of structural changes within a chromosome. So this is just a general overview between gene mutations and chromosomal mutations. So definition-wise, a gene mutation is just a change in the nucleotide sequence that is within a particular gene, whereas chromosomal mutations is an alteration of the nucleotide sequence of a segment of the chromosome, which actually includes a lot of genes. Okay, so the magnitude for a gene mutation is very small. However, chromosomal mutations can be seriously altered. Ability to correct gene mutations, it's much simpler than chromosomal mutations being altered. It is quite difficult to change chromosomal mutations. Then the number of genes that are changed in a gene mutation is just one gene, as I said before, and then chromosomal mutations end up with many genes being altered. Nature of this mutation with gene mutations, this is only a slight structural alteration, whereas chromosomal mutations are very like it depends on, it can be numerical or structural changes within that DNA or chromosome itself. Okay, and then the result is a simple gene mutation can have a low influence on different different phenotypes, but structural or variations in any number of chromosomes can lead to lethal diseases. So that's all I got for you guys. If you have any questions, don't hesitate to ask. We'll cover more during class next week if needed. Okay? Stay safe.